In this lecture, we're going to go over genetic drift. We've already done a little bit of that in class, so you're probably familiar with many of these things, but it's a good chance to review it. And then we'll talk a little bit more detail about what we can understand about populations and the study of natural populations. So before we look at the slides in the PowerPoint, I'd like to use our evolution simulator. You've got a link to this in the additional resources folder on Blackboard. You also have a link in worksheet number 3, and you're actually going to be asked to do some similar things in worksheet number 3. So as I'm demonstrating genetic drift here in the simulator, basically I'll be doing that last couple problems on the worksheet also. So you can use this as a really great guide for helping you to complete those problems on worksheet number 3. So keep an eye on that. Make sure you know of the due date. Worksheet 3 is due next week. So review that and make sure you keep up with those. So to do genetic drift, we're going to leave all of the fitness costs identical. So there's no natural selection pushing this one way or the other. There should be no random variation. And what we're going to show is that at a large population size, let's do, I don't know, let's say like 2 billion, something like that, that when we graph this, what we're going to see is there's going to be no change at all. So at these very, very large population size, there's very little genetic drift. Now, technically, to have zero genetic drift, you have to have an infinite population size. But you get to a certain point where it's effectively, its influence is effectively zero. It's kind of like saying, you know, how much money do you need to have before you're rich? And if you have $1 million, you're rich. If you have $1 billion, you're rich. And once you get beyond that, right, there's not much of a huge difference. So it's only when populations are small that we're going to have a major influence and impact for genetic drift. So let's bump this population down a little bit. Instead of 2 billion, let's say we just have 2,000. Excuse me. And when we only have 2,000 individuals, when we run this exact same simulation, now notice we didn't change anything. There's still no force pushing it one way or the other, but we're going to have random sampling error, which is essentially what genetic drift is. So notice that now, even though we didn't push it up or down, we ended up with a random fluctuation and it goes up. But if we ran it again, we're not going to be able to predict if we're going to end up at the same place, and in this case we wound up a little bit different. And so when we run this 10 times, we're going to have 10 different distinct outcomes. Sometimes it will go down, sometimes it will go up, sometimes it'll be pretty close to balanced, despite there being no force. So we could do that 10 times, and you're asked on the worksheet to do that with different settings, but you're asked to do that and determine how many become fixed in the population for one or the other. Now notice with 2,000 individuals, after 200 generations, we didn't have any of these 5 simulations where it fixed one allele, where we had lost one and fixed the other allele to 100%. But what would happen if we go down to a 200 population? So let's clear these simulations and try again. So with only 200 individuals, notice we lost the little a, so now we have only big A alleles. Now once that happens, there's no more genetic drift for this allele because we can't have random allele sampling and fluctuation. Now other alleles are going to be undergoing the same thing, and so this is only for this one site. And in fact, you can actually, if you want to, you can do 2 different genes at the same time. So let's do that just for fun, and let's look and see what happens. So here we have 2 of them randomly fluctuating. We fixed one allele for the big A allele. The little B allele became much more common than the big B allele, but didn't become completely fixed. And so we could do this, and notice that we're going to have very different outcomes, but this is key for genetic drift. Genetic drift impacts every single gene in the genome, right? Whereas natural selection might only be one or a small number of genes that are impacted by whatever environmental force is out there. But for genetic drift, it's everything. Okay, so for simplicity's sake, let's go back to one gene, and then let's do this just maybe like 5 times. So there we fixed the big A allele. We almost fixed it, went back down, then it fixed again. But notice we could just as easily fix the little A allele because there's not an advantage for one or the other. Genetics doesn't play a role here because we don't have any codominance or dominant systems going on. So if we ran this 100 times, we'd expect about 50 times to fix the big allele, about 50 times to fix the little allele, but there could be some variation. We wouldn't expect it to be significantly different. It's like saying if you flipped an evenly balanced coin 100 times, how many heads are you going to get? And you'd expect to see 50, but if you got 54 or 60, that might not be outside the realm of what you would consider probable based on that random distribution. Okay, so just be aware of this. Now, on the worksheet, you're given a graph where you don't have any natural selection, and you're asked to predict, is it going to go up or down? Now, hopefully your immediate instinct is, oh, I can't make any predictions. If it's going to go up or down, it's really random, and that's right. And so we can't make any predictions. However, I do want you to notice something here. When I have 200, even though I may not be able to predict the direction it's going to go, look at how randomly and how massively it's fluctuating. This one actually ended up just by chance kind of back where we started, or at least close to it. But notice how randomly, massively it's bouncing up and down. So fluctuation does tell us something about the population size. Whereas if I have 20,000 individuals, even though again I can't predict now, is it going to go up or down, I can predict the amount of that generation fluctuation is going to be much, much lower. So less random fluctuation from generation to generation. And again, I can't predict if it's going to go up or if it's going to go down, but if I did this 10 times, I would expect a much narrower range at the end of 200 generations because we have less random fluctuation. Whereas with only 200 individuals in the population now, can't predict up or down, but I can predict, hey, it's going to be crazy, right? It looks like the stock market recently, up and down and up and down. But with small populations, because of this random sampling error, we're going to have those random fluctuations. So although you may not be able to predict direction, you could tell me something about the size of the population based on how jagged and how extreme these generation to generation fluctuations are. So keep that in mind when you're doing the very last problem on the worksheet. That's key, okay? So let's take a look at the PowerPoint. So now you know about genetic drift, and if I looked at these, there's information here about the population size, but even if I didn't tell you these population sizes, say I'm going to do 10 random simulations. The blue one's just one of the simulations that's highlighted. You could say, oh look, this population's much smaller than this population because of that random fluctuation. Okay? So the effect of sampling error on evolution is dramatic, particularly in very, very small populations. And not only that, but it impacts every single gene in the genome, right? So that can be massive. If we have a little tiny population, right, then we're going to see random fluctuation from one generation to the next. We're going to lose a lot of genetic diversity as some alleles get randomly fixed. And not only that, but if it was small enough, then genetic drift could potentially even overcome the effects of natural selection. So let's do that. So now I'm going to put a little bit of natural selection here. Let's say the fitness of that one's 0.9, and we'll say maybe 0.95 there. So some natural selection. Actually, it's a fairly strong force of natural selection. So in a large population, we're going to see only natural selection. And it's going to very predictably, simulation after simulation, do the exact same thing. So very little genetic drift. We'll lose the big allele because it's worse than the little allele. Nice. Hopefully that makes sense to you, okay? But what happens if I add in a big component of genetic drift? Let's go back to 200. In fact, let's make it even worse. Let's do 100 individuals in the population. Now, we should see it going down, and natural selection is going to continue to force it down with the same force. So, it went down. But why did that one go up? Well, in this case, the random fluctuations are so strong that just by chance, we may actually lose slightly beneficial alleles. Now, if we make this a stronger impact, then despite there still being random fluctuation, that force is so strong it just slams it right down, and so the random fluctuation does not have much of a chance. But with, significant but lesser natural selection impact, we're going to have this random fluctuation. There is a force pushing it down, and so we get a majority of them, maybe 9 out of 10, or maybe 8 out of 10, that are going to lose the detrimental allele. But every once in a while, there we go, there's that one where the random fluctuation pushed it in the wrong direction. Now, wrong's maybe not the right word, but put it in a direction that counters natural selection. So just be aware of that. And these forces are genome-wide, so they can be massive and even really, really be the major force of evolution on little tiny populations. Okay, so we're going to do some definitions here. Demography, in general terms, is just the study of populations. And there are human demographers that are looking at social and cultural and political trends also. But when we say demography, we mean natural populations. And so we're interested in population history. So this might be studying the populations over time, right? Or looking at impacts of predators. Anything that impacts population could be put under this umbrella of demography. And so one scientific discipline within demography is biogeography. So demography is simply the study of populations. Biogeography is the study of populations and where they are found and according to their landmass. And this can be done with extinct things using fossil records. So here is a very generalized map of where we find certain types of fossils. And this is actually one of the things that led scientists to the theory of plate tectonics, because they were finding similar or even identical fossils in Africa and South America and across all these southern continents. So that would be one way we could do biogeography. We could also do it using modern populations by defining these biogeographic realms or ecosystems. So we've got basically the far north, right? The Arctic Circle. We've got the taiga, which is the large band of conifer forests that occur in northern Europe and America and across into Siberia. And so that's going to be different, and although we might find very similar things if we travel 100 kilometers in this same biogeographic area, if I traveled 100 kilometers to a new biogeographic area, I might see very, very different flora and fauna. Okay? And then phylogeography is actually just kind of a subdiscipline of biogeography, where now we're going to add our understanding about relationships, about phylogeography. So we look at organism distribution, we look at the history of those land masses, and we throw in our understanding about phylogenetics to tell us something about the history of those organisms. And we'll look at a couple specific examples here in Unit 3.2b. Okay, so just know those definitions. Another really key critical definition is heterozygosity, and technically this is heterozygosity with a capital H. Heterozygosity is a measurement of overall genetic diversity in a population. And so we could do this by hopefully using more than one gene. Maybe we sample 5 genes, and then we look at how many individuals are heterozygous for gene 1, gene 2, gene 3, gene 4, and we come up with an estimate of the overall heterozygosity. In large, healthy populations, heterozygosity is going to be pretty high, large. Okay? And so if we see overall heterozygosity dropping, and remember, if this is really a good estimate over the entire population, then this is showing that there's something going on. And the most common, easiest explanation for why heterozygosity would go down is genetic drift. So this is a sheep's head fish, the population off the coast of California. They've been under some fishing pressure, so their populations have dropped. And so we can see how severe that population drop is. Even if we don't go out and count fish, if we just look at the heterozygosity, then we know the population is dropping. So it becomes a measurement that can kind of stand in for maybe other measurements like overall population size that might be a little harder to do. Okay? So how small does a population need to be for genetic drift to play a role? Well, technically genetic drift plays a role in all populations, but it's really not until it gets down into the neighborhood of tens of thousands or thousands that we start to see a significant drop. And that may be different for different populations. Some populations naturally go through these boom and bust cycles where we get very small population sizes. Okay? But it plays a role in all populations. The smaller the population is, the more severe and the more extreme that role of genetic drift is. Okay? And so we are very interested in population size as we look at organisms. And so whether we do this and estimate population size based on heterozygosity, which we actually can do if we use the right procedures, or if we're actually going out and counting animals, we want to know how big populations are. That's critical for understanding the history and even the future of healthy biological populations. And in fact, there are actually 2 numbers that you need to be aware of. Census population size is an estimate of population size based on just counting all of the individuals. In 2020, the U.S. is doing a census, which is an estimate, an attempt at least, with some extrapolations made to try and count every human in the United States. And then we can do things like define and design public policy moving forward, allocate representatives to Congress, right? That's one thing that's done based on population size. So that's a census population size. Just count everybody. But in biology, we are actually much more interested in this other sign, which is usually designated by an N subscript E, which is the effective population size. And I put a sea turtle up here to represent that. If we went out and counted every living sea turtle, that's actually not a good reflection of how many individuals get to reproduce, right? So we might have hundreds of thousands of juvenile sea turtles that are out there as they hatch and are going, but maybe only 1 in 100 or 1 in 50 survive to reproductive age. So census population size is everyone. Effective population size are only those that are going to get to reproduce. And for some populations, like the human population, those are pretty close, but by definition, in real populations, N subscript E, the effective population size, has to be less than the real population size. In ideal situations, they could be equal. Effective population size can never be larger by definition than census, but in real situations, you're always going to have a few individuals that die. And so we're never going to have those numbers be equal in real situations. And in some populations, it's extreme, right? So we have these populations where their strategy is to make thousands upon thousands of little tiny babies and hope a few survives. So they may have a very large census population, but a very small effective population size. And so even though I've only put 3 numbers here and we're not going to fill them in, we could make an incredibly long list, right? We could talk about infant mortality rates. We could talk about predation. We could talk about, just random accidents. We could talk about diseases. We could go on and on and on about why these numbers are different. So just recognize, and I don't expect you to know any 3 particular, just recognize there's a long kind of a laundry list of why these numbers are different. Make sure you know what they are, and that it's the effective population size that's the one that's really, really important and critical for biologists. Okay, that's the end of this unit. Actually, let's just do this one last thing. This kind of relates to the next thing going forward, but it's worth talking about. So if we have the exact same experiment set up in 5 different places, so these are 5 hypothetical islands with the exact same founding population, everyone's a heterozygote. Obviously not a very realistic situation, but we could maybe set this up experimentally and then look to see what would happen. We would, if there's no natural selection at all, every island is identical size, identical environment, we would expect to have eventually differential outcomes and fixation of one allele or the other. Given enough time, eventually one allele will become fixed. And the time to fixation is simply dependent on how big the population is. So if it's fluctuating like crazy, eventually we'll fix either one allele or the other. We don't know which because there's no advantage for one or the other, but we'll do that. And so even though this is an artificial setup, we do have some real-life biogeographic examples where we can see this, so it's kind of cool. So for instance, during the Ice Age, ocean levels were much lower, and as oceans have risen, different land masses that were once one island with a single population have become 2 islands. So looking at this population of lizards in the Galapagos Islands, we can treat the Santa Cruz population different from the Isla Santiago, and by looking at the history, we might even know when they were broken up. And then we could see that if there is variation between them, it may be due to natural selection. Maybe the island conditions are a little bit different, but it's probably more likely, because these islands are close to each other and still very similar, that differences we see between these now separated populations are due to genetic drift and not natural selection. So we could study that, look at the effect, come up with an estimate, see if it matches what we understand about the history, and do some really cool and interesting studies based on the demography and the genetic diversity of these populations. Okay, that's it for genetic drift. Look at 3.2b. That will be the next one for you to review.